Antibodies that circulate throughout the blood and lymph are called immunoglobulins. 5 types of immunoglobulins have been identified, each with a particular role. Each antibody has at least 2 antigen-binding sites. An antigen-binding site is specific to only a small portion of the antigen, called an epitope. Antibodies lead to the destruction of antigens through several mechanisms. Since each antibody has at least 2 binding sites, one antibody can bind to identical epitopes on 2 different bacterial cells or viral particles. Different antibodies will bind to different epitopes on each antigen. This links the antigens together. Eventually, they form a large clump that makes the antigens more easily identifiable to macrophages. The binding of antibodies to the surface of a pathogen can also activate the complement proteins. Complement proteins attack the pathogen's plasma membrane by forming a pore that allows water and ions to enter the cell, causing it to lyse. This is similar to the role of the complement system in nonspecific immune defense, except that here the proteins are recruited by the bound antibodies. In general, the specific responses of T cells, B cells, and antibodies are balanced to protect the body against many diseases. However, sometimes that balance can be upset.